Hi, so I'm Matthew and I am the team lead for the BioImage Archive at EMBL-EBI. And I'm going to tell you a little bit about the archive, trying to focus particularly on aspects of what we do that are relevant to image analysis and getting understanding out of images. So the BioImage Archive is one of the newest data resources at EBI. And our mandate is to take imaging data, store it and index it and give it back. And we do this really for four main reasons. One of those is reproducibility. So lots and lots of results in biological sciences are based on imaging data, and really this whole meeting is about the process of how we get knowledge, how we get information out of images. But when we come to present our work and when our work forms part of published pipelines, published manuscripts that support hypotheses, we need to be able to show how we got to those results, and we need to be able to track back to the original raw, preferably data underpinning those results. So that's one of the things that we do. We take that raw data to enable reproducibility. We also take what we sometimes call reference data sets, so particularly complete or important data sets that are of special importance to the scientific community. For example, cell atlases, where the data sets take very large amounts of time to generate and curate, so we have to kind of make them available for as long as possible. But we also take data for reasons of really enabling new science, not just reproducing existing science. And that new science occurs through two main routes, one of which is where we can derive new scientific results from existing images, from existing image data. But another route is through the development of new image analysis tools and techniques. So really we're here to try to support the work of the many sort of developers and scientists here who are building new pipelines, new tools, through provision of reference data and benchmarking data, and also training data in the sense, not just in, for example, model training in AI or machine learning, but also training for training events. And to meet these goals, we take data, and we take data really from any scale of imaging, from molecules through to organisms, as long as those data are related to results in a publication or have some value beyond a single experiment. So in a sense, the reference data sets that I was talking about before. And the reason for those constraints is we're not trying to be the image data management platform for the whole world, for everything that comes out of an imaging instrument. That's just not practical. Our role is open access to published data. And we will take data in actually any format, at least at the moment. The other constraint being is we can't take patient identifiable medical data because we don't have the anonymization infrastructure. How does submission to the archive work? Well, you can find out more detail on our website, which I've linked here, ebi.ac.uk/bioimagearchive. And that will give you a lot of detailed information about how the submission process works. But thinking about it at a high level, there's really four steps. There's organising the data that you want to submit. So that might, in some cases, be just raw data. If you're publishing an analysis tool or pipeline, it might be raw data plus analysed data plus analysis results. We will take all of that. You then register an account with us. You transfer data over to EBI, and there's different ways of doing that depending on the scale of the data that you're working with. And then you fill in some metadata, and I'll tell you a little bit more about that later on to give some context to the data set. And most of those processes take place through a web graphical interface, so it's relatively user-friendly. There are other ways to submit data for unusual types of submissions. And when all that's complete, we will give you an accession identifier, a permanent identifier for your data. And you can also choose when you'd like the data to be released to the public. Often you'll want to do that say when a manuscript is released or a preprint. We can also provide anonymized links for reviewers if you want to be able to share the data with specific sort of groups or people before full publication. And the process can be as fast as a day or two for smaller data sets. For larger data sets, it tends to be the data transfer process itself that's the rate limiting step. What sort of data do we have so far? Well, we have lots of light microscopy, because it's one of the most common imaging techniques. Here are some examples chosen to a large extent because they're visually appealing. And with the images themselves, we also have the metadata that I mentioned before. So we have the information that gives context to the data. So why were the data being captured? What was the purpose of the experiment? What was being imaged in terms of, in this case, it was human cells. What type of experiment was it? And then there's more detail about the protocols and how the images were captured. We don't just take human data, we also take data from other species. So here's a plant example. And we take data from lots of different experiment types. So here is a high content screening example. And I picked out a high content screening example to show that for data sets like these, we have a lot of information associated with each image, because this is a compound screen, and in a compound screen, the things that are important are information about the compounds themselves. So what compound corresponded to each image in the data set, and for those compounds, say the formulae, links to identifiers, ways in which you can associate the images with other information. But beyond the more common imaging types, we have a whole range of different types of imaging, because we're a very broad-based archive. We have photoacoustic imaging and micro-CT and ultrasound and all sorts of interesting things. In terms of data volume, or at least numbers of data sets, the archive launched officially in 2019. EBI was taking imaging data on a small scale before that, which is why the graph goes back a little back before 2019. But really, sort of development effort has kicked off in the last 6 to 18 months, which is why the volumes of the data we have been growing extremely rapidly. This is sort of near exponential, and especially the last few months, the number of data sets we hold has grown very rapidly. We're now well above 200 and counting. Briefly, before I finish, what does the future hold? What are we working on at the moment? Well, I've mentioned metadata a few times. Metadata, information about data, is really important, as those of you who do image analysis will be very aware that, for example, if you want to make measurements in physical scales, you need to know the conversion rate of pixels or voxels to physical length or volume or area, I suppose. But there's lots of other metadata that's important to capture about images, from why were they being captured in the first place? Were they part of a larger study? What exactly was being imaged? What type of instrument or instrumentation was used to capture the images? Again, for this context, if analysis was done, how was that analysis done? Links to software packages, parameters. All of this is important to capture. And the EBI was involved in coordinating a community effort to try to come up with a standard for the types of metadata that should be associated with images. And really this model, which is called REMBI, for recommended metadata for biological images, groups the metadata into different categories. And not all of these things are necessary for every imaging deposition. For example, correlation doesn't apply except in correlative studies, and analysed data is more for presenting analysis results. But this provides a framework to fit the metadata in, and we're working on implementing this in our submission and sort of data exploration process. We're also looking very hard at data formats and ways to sort of make sure we get the most use out of our storage. It comes up in other contexts in this meeting, but the next generation file formats, the development of which has been led by the OMI consortium and others, are really important to us, because those are really designed for sharing large data sets publicly in ways that are sort of friendly to cloud analysis and interactive visualization, like this nice JavaScript viewer. This is Visar, which you may be familiar with, shown in the middle panel. So these will let us get more out of the data we have and let you explore that data in new and interesting ways. So to stick to time, I will finish there, just to thank on the left side several of my colleagues and team members at EBI on the top right, advisory group, and our funders, both current and previous funders, without whom none of this work would have been possible, and we wouldn't be able to keep running the archive. And thank you very much to all of you for listening.